type of motif is about 30 micromolar, so it's a very weak interaction, or a relatively weak interaction. Each of these represent a different pull down in which you've done an alanine scan through the motif. And what Brian and Mashida Alam, an undergrad in the lab, found is that the motif has important binding determinants at either end. So there's an N-terminal portion of the motif where substitutions at this part really disrupt binding. We're actually able to insert up to 4 amino acids in the middle of it and not affect binding. So we think there's a flexible linker in the middle. And then there's a C-terminal motif, which has this YQFP. And more about this in a moment. If you have a docking motif present, you see robust phosphorylation of a fragment of ACE2 in vitro. But if you mutate the docking motif, and again, this is about a 30 micromolar interaction, you actually have almost a 100-fold effect on phosphorylation of this fragment in vitro. Interestingly enough, I don't have time much to talk about this, but binding to just the docking motif activates the kinase as well, for autophosphorylation at least. So that's great. And James Hellman was talking earlier in the meeting about how the biological power of twofold effects. So here's something like a 100-fold effect. So is there a biological output? Well, disappointingly enough, it was actually pretty modest. So here's looking at a failure to separate. So here we have cells growing in culture, and a tendency towards this end of the graph means that the cells aren't separated. If you delete ACE2, the cells don't separate. Wild-type cells, you see them mostly as 1 to 2 cells in culture. Occasionally they stick together, you get 3 to 4 cells. If you mutate this docking motif, you see a pretty modest effect, right? Really not much. Now, however, if you put this docking motif mutant of ACE2 into cells that have a weakened allele of CDK1, so this is an analog-sensitive allele of CDK1, without this drug present, you see this allele by itself has almost no effect on cell separation, but in the presence of the docking motif, you see a major effect on cell separation, right? So what this means is that this docking interaction is not essential for this regulation to occur in vivo, but it confers robustness, defined as the ability to maintain a consistent output of the system in the face of widely varying input in terms of kinase activity. You see this more dramatically if you look at transcription of CTS1, an ACE2 target gene. Wild-type cells have about this level. If you have the analog-sensitive mutant, you see a modest decrease, or a modest decrease with docking motif mutant, but you see a great decrease if you combine the docking motif with this weakened allele. All right, so we took a look at this motif and focused on the C-terminal part of it. And what we found is that a number of other proteins have this FXFP or YQFP motif. And in fact, SSD1, the other known target of CDK1, has this motif. And in SSD1, just FXFP is sufficient. So there are 2 of these motifs here, FKFP. And if you mutate those, here you see phosphorylation of a fragment of SSD1. And with those sites mutated, you see very poor phosphorylation of that. And I'll just summarize our findings to say that, like with ACE2, this docking is not essential for the kinase to regulate the protein in vivo, but rather it confers robustness on this regulation in vivo. All right, so now for something a little different. Now just put aside everything I've just told you, and we're going to take an unbiased approach to find these kinds of motifs. So the idea here is that a lot of the action for these short functionally important motifs are going to be on the surface of proteins, as Martha was kind of introducing to you. And the idea is that a lot of loops on the surface of proteins are actually not structured. They're disordered inherently. And as such, they are rapidly evolving quite often, except for where there's a functional motif present. And the idea is functional motifs present in unstructured regions at the surfaces of proteins are going to have a lower substitution rate because they're constrained and indel rate than the surrounding context, right? And so what Alan and Alex in their group in Toronto did was they applied a technique called the phylo-HMM. It's a Markov model-based approach in which you take alignments of orthologs of all yeast proteins, about 5,000 alignments, and then what you do is you compute a local substitution rate, local evolution rate within a window, accounting for amino acid substitutions and also indels, and you treat indels as blocks. And then you assign a probability that any one amino acid in that sliding window is evolving at the window rate of evolution or at a slower rate of evolution, right? And what you get are a bunch of conserved segments, and then you remove, by a computational approach I can tell you about later if you're interested, the structured regions of proteins. And what you get out is about 20% of the protein. About 20% of the yeast protein is unstructured inherently by these methods. And you get about 7,600 conserved segments. And then what you do is you take those segments, you align them, computing pairwise distances, and you cluster them, and you get about 282 clusters. And these clusters include many very important motifs, right? So this includes things like serine proline, and a majority of these are actually cell cycle regulated proteins. And the statistical enrichment is very high. You get KR, NPF motifs, you discover the KEN box, HDEL, a bunch of functionally interesting motifs in this, as well as many, many motifs that are unknown. And this approach identified the CDK1 docking motif. So a cluster of 6 proteins. 4 of those 6 had previously been known to physically interact with CDK1, so the probability of that happening by chance is about that, which is very small. All of these proteins are phosphorylated at CDK1 sites in vivo. About 130 proteins are known to be phosphorylated at that site by mass spec. So the probability of that is also very low. And so what this overall does is give us a picture of an integrated system of substrates. Docking plus consensus gives you 2 known substrates and other substrates as well. There are localized decisive cytokinesis and polarized growth. This guy's involved in poly-A, and it's important for cell separation. And also ZDS1 and 2 are things involved in checkpoint and approach GPase function. Additionally, there's a non-docking way to look for targets, and that includes things in polarized growth. Finally, and I won't mention it, but this docking motif, for those of you who know about MAP kinases, is the YXFP, FXFP is what MAP kinases dock to. And we found that Fuz3, at least in vitro, will bind to those exact same docking motifs. These are the people who did the work. I focused on work by Brian Ye, but importantly, Ellen Mazanka contributed functional understanding of the system, as did Jackie Jansen, who's sitting back there in the back.